In this video, you will learn about benzonase treatment of the sample, buffer preparation, and DCS cassette assembly. To maximize performance of the Curate Cell Processing System, additional citrate must be added to the leukopak at the time of collection, and the leukopak must be stored overnight on a bag rocker at 2 to 8 degrees Celsius or shipped cold for next day delivery. Using the leukopak volume to be processed, calculate the required volumes of additives for proper treatment. Refer to the Instructions for Use for recommended concentrations and materials. Under aseptic conditions, remove the cap from an empty conical tube and dispense the required volume of 250 millimolar magnesium chloride into the falcon tube. Discard the pipette tip. Adjust the pipetter to the required volume of benzonase nuclease. Dispense the benzonase nuclease into the conical tube that contains the working volume of magnesium chloride. Use the pipetter to adequately rinse the pipette tip. Connect a 2 1/2 inch 18 gauge needle to a 20 milliliter syringe. Immerse the tip of the needle into the solution inside the conical tube and withdraw. Disconnect the needle from the syringe. Use the transfer set's lure adapter to connect the syringe to the leukopak bag. Release the clip on the transfer set and push the solution into the sample bag. Gently withdraw the plunger and push the fluid back into the bag. Repeat this step 2 additional times to ensure proper mixing of the leukopak and additives. Close the clip on the transfer set and remove the syringe. Place the cap on the transfer set's lure adapter. Carefully mix the leukopak bag by inversion 3 times. Place the leukopak bag on a rocker at room temperature while completing other preparation steps. Next, prepare the priming solution bag. Gather the listed supplies into the hood to be handled under aseptic conditions. To begin, remove a fluid transfer set from its packaging and close the clip near the spike end of the adapter. Remove the spike port cap on the 250 milliliter 0.9% saline bag and insert the transfer set's spike into the exposed port, twisting slightly to maneuver it in all the way. Next, connect an 18 gauge needle to a 20 milliliter syringe. For safety, be sure to leave the sharp end of the needle covered until it is time for use. Obtain the Curate priming reagent bottle. It contains a transferable QR code label to be placed on the priming bag after preparation. Carefully lift the center disc of the aluminum crimp seal to expose the septum. Pierce the rubber septum, and with the needle still inside the bottle, slowly turn the bottle upside down and adjust the end of the needle so its tip is immersed within the liquid. Pull back the syringe stopper and withdraw 15 milliliters of the Curate priming reagent. Invert the bottle and carefully detach the syringe while leaving the needle inside the septum. Remove the transfer set's cap to expose the lure connection. Connect the syringe, open the clip, and slowly inject the Curate priming reagent into the bag to prevent any frothing. Gently withdraw the plunger and push the fluid back into the bag. Repeat this step 2 additional times to ensure proper mixing. Close the transfer set's clip and disconnect the syringe from the adapter. Replace the cap and gently mix the priming solution by inversion 3 times. Once the new solution is prepared, remove the transferable QR label from the reagent bottle and place it onto the now prepared bag. This QR code will be scanned during the Curate system's accessioning or loading process and will ensure traceability. Next, you will learn how to prepare the run buffer, which consists of 4% human serum albumin in Plasmalyte. Again, gather all materials to be handled in a hood under aseptic conditions. Begin by removing a fluid transfer set from its packaging and closing the clip near the spike end of the adapter. Remove the 1-liter Plasmalyte bag's spike port cap. Carefully insert the transfer set's spike into the exposed port, twisting slightly to maneuver it in all the way. To prepare 4% HSA in Plasmalyte, inject 191 milliliters of 25% HSA into the bag, preparing a total volume of 1,191 milliliters. Alternatively, remove 160 milliliters from the Plasmalyte bag and replace the volume with 25% HSA. Connect an 18-gauge needle onto a 50 milliliter syringe and gently pull back the plunger to withdraw 50 milliliters of air. Pierce the bottle's rubber septum and slowly turn the bottle upside down. Inject the air into the bottle. This will prevent pulling against a vacuum. Release the plunger and allow the fluid to enter the syringe. Pull back the plunger as needed to withdraw 50 milliliters of 25% HSA. Keep in mind that the albumin is viscous and this process may require additional time. Remove the syringe from the needle and connect to the transfer set via the lure adapter. Undo the transfer set's clip and slowly inject the HSA into the bag. Gently withdraw the plunger and push the fluid back into the bag to ensure proper mixing. Repeat this step a few times as needed. Make sure to close the transfer set's clips before undoing the syringe. Replace the cap and gently mix the Curate run buffer by inversion 3 times. Repeat these steps until the required volume of HSA is added to the Curate run buffer bag. Next, you will learn how to prepare the diluent bag. The Curate system requires the use of a 250 milliliter 0.9% saline bag as diluent. Prepare this bag by first removing a fluid transfer set from its packaging and closing the clip near the spike end of the adapter. Remove the saline bag's spike port cap and carefully insert the transfer set's spike into the exposed port, twisting slightly to maneuver it in all the way. To fully assemble a DCS cassette, begin by organizing the listed items inside a hood. While Charter Medical expansion bags are recommended for product and waste bags, any sterile bag with weldable tubing can be used as long as the bag volume matches the expected output volumes. The sterile, single-use DCS cassette is provided in a convenient carrying tray. Place the cassette in the hood to begin assembly. The Benzonase nuclease treated sample and the buffers prepared earlier will be connected to the cassette. Begin by removing the DCS cassette's Tyvek lid. Inside the packaging, you will find color-coded QR labels corresponding to each of the bags. Place the color-coded QR labels on each bag. Note that the Curate priming solution bag will receive two QR labels, white for the priming reagent bottle and yellow for the priming solution. Place the blue Run Buffer QR label on the 4% HSA in Plasmalyte bag. Next, place the purple Diluent QR label on the 250 ml 0.9% saline bag. Place the red Sample QR label on the Leukopak bag. Remove the 500 ml product bag from its packaging and close all tubing clips. Place the green Product QR label on the product bag. Lastly, remove the 1-liter waste bag from its packaging and close all tubing clips. Place the black Waste QR label on the waste bag. Before proceeding, close all 7 color-coded clips on the DCS cassette. If necessary, move the clips away from the spike end to ensure that there is enough tubing length remaining for welding. Note that failure to close the clips will cause improper flow of fluid into the cassette, leading to a failed process. To continue with sterile weld connections, remove the spikes from their retention features and use a sterile tubing sealer to seal the red, purple, blue, and yellow cassette tubing immediately below the attached spikes. Note that shortening and/or lengthening the cassette tubing may lead to a failure. You may detach the sealed-off spikes by pulling the sealed tubing sections apart. After this step, you may remove the packaging tray and other supplies from the sterile environment and continue with welding. Begin by removing the looped tubing from the inner tray's central retention features and securing them to the retention feature on the right-hand side of the tray. This will allow better access during loading. Remove the tubing with the green clips from the tray's retention features. Sterile weld the 2 tubes marked with green to the product bag with the green Curate QR code label. Do not weld the green tubing to the product bag's tubing meeting at a Y junction. Gently roll the welded tubing in between your fingers to open the weld. Place the product bag on top of the cassette. Repeat by removing the tubing with the black clip from the retention feature and welding to the waste bag with the black Curate QR code label. Place the waste bag on top of the cassette. Rotate the packaging tray for better access during the next 4 steps. Remove the inline filter from the retention features by lifting the filter from the top and bottom ends. Do not pull the tubing to release the inline filter. Weld the sample bag tubing to the stub of cassette tubing red clip that remains above the filter where you remove the spike, and place the sample bag on top of the cassette. When welded correctly, the filter should be within 10 centimeters of the bag. Place the sample bag on top of the cassette. Remove the diluent tubing purple clip from the retention feature and sterile weld to the diluent bag purple Curate QR code label. Place the diluent bag on top of the cassette. Remove the run buffer tubing blue clip from the retention feature and sterile weld to the run buffer bag blue Curate QR code label. Place the run buffer bag on top of the cassette. Lastly, remove the priming solution tubing yellow clip from the retention feature and sterile weld to the prepared priming solution bag yellow and clear Curate QR code label. Place the priming solution bag on top of the cassette. The cassette is now ready for use. For more information, scan the QR code to download the instructions for use or contact Curate Biosciences by email or phone.